Hello, everyone. I hope you guys can hear me. My name is Ahimsa Campos Arceiz, and today I'm representing Integrative Conservation, which is an academic journal of which I'm one of the co-editors-in-chief. And we are very honored to work with the Snow Leopard network on this collaboration to do this series of 2 webinars. At least at the moment, the plan is to have 2 sessions. One is today, another one will be on the 12th of May. On the snow leopard as a large cat with high potential for conservation success. So then this is the plan for today. These are the speakers. They will be introduced later. Let me do a bit of a brief introduction to integrated conservation. So then integrated conservation is a relatively young journal. It was launched at the end of 2022, and we are now in our 5th volume. And we publish 4 issues per year. It's a conservation journal. It's highly integrative. So then basically we are keen on publishing conservation work from any system, from any geographical region, at any level of a biological organization. And we aim for interdisciplinary content. So then things that integrate multiple perspectives, multiple dimensions, understand that conservation problems are wicked problems, and we don't want to portray like simple stories or less simple solutions. We aim for very high quality work. Of course, this journal belongs to the Xishuangbanna Tropical Botanical Garden, which is part of the Chinese Academy of Sciences, which is the largest scientific organization in the world. And as such, we have a very strong academic support behind us. The journal is published in English with a Chinese abstract and some Chinese title and other contents. We are very keen on facilitating access to people from different languages. So then, for example, we do in-house English editing, and of course, we welcome publications, abstracts, practitioner points, or even the whole paper translated to the local language. So then it can be better conveyed to practitioners and policymakers. And very important, we are open access, we are free to read, and we are also free to publish, at least for now. So then until now, we don't have APCs, and we intend to continue without APCs for as long as we can. So then I highly encourage you guys to think, if you have a cool conservation work that you want to publish, please consider our journal. We publish special issues, and actually the event that we're doing today is one of the outcomes of the special issue we published before on snow leopard conservation. We published 8 papers on snow leopard from different countries on a different range of topics and in different formats, going from editorial to commentary, research paper, and review. And then we are now calling for papers on 2 different topics on special issues. One is on man and biosphere, or it's called Balancing the Needs of Man and Nature, Biosphere and Beyond. And also we have a special issue on the biology and conservation of Asian turtles and tortoises. So if you're working on these topics or know anyone who is working on these topics, please consider submitting something. If you have an idea for a special issue and then you would like to discuss with us, we very much welcome ideas. Happy to discuss with you. If you want to access the journal, this is an easy way. You can just scan this QR code, and then just remind you that we'll have a second event on the 12th of May. It will be at 4:30 p.m. Beijing time, so it's about 1 hour later than now. And at that time, we will have, again, Li Yuan, who's going to be facilitating today's event. We'll have Xiaolin Yun. Both of them are from Xi'an Jiaotong Liverpool University here in China. And then we'll have Sandro Lovari from the Maremma Natural History Museum as one of the speakers. And then, without any more delay, so then I will go back to today's moderator, and we'll hear from this exciting presentation. So thank you very much. If you have any question, please feel free to contact myself or the editorial office that you can find information in our website. Thank you very much, Dijon. The floor is yours.
Thank you for Ahimsa to introduce about the integrity conservation. And I am Juan Li. I'm an assistant professor from Xi'an Jiaotong Liverpool University. And I'm also pretty honored to be the guest editor of this snow leopard special issue. So as Ahimsa just mentioned that Our special issue have 8 papers, and which cover 3 different themes, including establishing baselines, advancing methods, and transboundary conservation. Today, we're pretty honored to have 2 of the authors from the paper published in this special issue to talk about their pioneered research. So I'll introduce the first one. So the first one is Ms. Charu Sharma, and she is then a research assistant with the High Altitudes Program at the Nature Conservation Foundation, India. And she is an early career researcher whose work focuses on using camera traps and refined methodologies to study wildlife in mountain ecosystem. Her research interests including population dynamics of mountain ungulates, spatial ecology, and evidence-based conservation. And she's passionate about bridging scientific inquiry with collaborative, community-driven conservation efforts. And Charu will present the paper titled A New Dawn, Population Baseline of Snow Leopard and Other Mammals of the Quechua High Altitude National Park, India. And Charu, the floor is yours.
Hello, everyone. A very good afternoon. I am Charu Sharma. I work with Nature Conservation Foundation India. And today I will be describing you about our work on population baselines of snow leopards from Kishtawar High Altitude National Park in India. Yes. Before I delve into the details of the study, there are a bunch of people I would like to thank who have contributed immensely to this work and to the study also, all our partners, collaborators, funders, and everybody on the screen, and many more people. Thank you so much, Munib, as well, for giving me this opportunity. Right. So we all are well aware that snow leopards are a great flagship species for the high Asian mountains and the Central Asian mountains in the world, and they are also threatened. However, a very small proportion of their population has been globally systematically sampled to arrive at reliable estimates. And population baselines are necessary to inform about the current status of these threatened species to then further guide conservation measures. Also, Himalayas, like we can see in this map, Himalayas are also a highly global biodiverse region and an important snow leopard habitat. However, a large part of the study on snow leopards has focused on the trans-Himalayan habitats in India, whereas the greater Himalayas have been understudied, less looked upon, whereas they are also a great potential or good potential snow leopard habitat. To the left, you all can see how the trans-Himalayas look like, and the right side image depicts how the greater Himalayas, which are slightly more diverse and transcending elevationally. With this motive, our aims were to establish a robust baseline of snow leopard population in the Kishtawar High Altitude National Park, and also understand the relative abundance and diversity of all these high altitude mammals found in the region. The Kishtawar High Altitude National Park is located in the Western Himalayas of India, in the eastern part of Jammu and Kashmir. This is also a unique biogeographic region since it spans from the greater Himalayan regions and is surrounded by the neighboring trans-Himalayas. We'll see in the coming slides. And there are these 4 larger river catchments in the region. There is the Renai-Madhwa region. Then there is Kiar Valley. There is Nant Valley. And then there is the Kibber Valley, which is a very small catchment. Our sampling, or we studied this southern part or the Dachin region of this national park. And like I said, because it's the greater Himalayas, we see elevationally, once we go inside the valleys, the landscape and the habitat changes dramatically. How Kishtawar National Park changes is in the lower areas. Elevationally, this region spans about starting from 2,000 meters, going as high as 6,000, 6,500 meters. And once you start entering the region, it's filled up with these coniferous forests, the pine forests, transcending into the alpine meadows, the rocky steppe ecosystems, and the higher parts or higher areas have permanent glaciers in the landscape. Along with this, most of the villages are located outside on the boundaries of the national park. The image on the left is a reference of how the valleys look like. And they practice agriculture for subsistence. There is also a form of seasonal land use in the national park, where the nomadic herders, Gujars and Bakarwals, visit the high pastures in the landscape seasonally to graze their livestock, primarily sheep and goat. Jumping into what we did for this study, we placed camera traps to arrive at snow leopard estimates using the spatially explicit capture-recapture framework. A total of 44 camera traps were placed. We placed them during April to June period, ensuring that it's small enough for a closed population, assuming no movement in and out of the population for a period of 45 days. We sampled 3 valleys, and we tried maximizing our placement of camera traps where we could see potential snow leopard signs, ridge lines, cliffs, and microhabitats where snow leopard would come and visit, so that we can get as many captures as possible. Also, in the study design, like we saw in the map, the camera traps are placed slightly in a clustered manner because of the terrain of the region. And clustering also thereby helps us ensure that we get enough recaptures. And the overall study area, or the study expanse and coverage, was large enough to cover multiple snow leopard home ranges. For the analyses, we use the multiple likelihood SCCR framework, a single session SCCR model where we assume the density, the detection probability, and the sigma, an indicator of the home range, all these parameters to be constant in this area. We chose a habitat mask with 18 kilometers buffer, 1 kilometer spacing. And like I described, the national park has a huge elevational gradient, but we clipped our habitat with respect to the knowledge from the landscape to the snow leopard occurrence range, which was 3,000 to 6,000 meters. For all the other detections and other mammals that we got in our camera traps, we arrived at relative abundances, which was a ratio of the number of encounters in front of the camera traps divided by the number of camera trap days, our total effort, times 100. Right. So what did we find? Before also jumping into the results, I wanted to discuss a bit about how we process the data. Once we retrieved the camera traps, we classified or categorized all the kinds of wildlife that we got, filtered out our images. And for snow leopards, individual identification using their rosette patterns was done, since it's a marked species and a marked animal, in a 2-step process where where 2 observers individually identify snow leopards, and the discrepancies are then resolved to ensure that the ID is carried out as correctly as possible. And then that information went to feed into our models. And we found that snow leopards are moving across all our valleys. Out of the total 44 traps, we found them in 9 locations, 15 detections, and 4 unique individuals were identified from our data set. Right. And the density estimate for this region also came out to be comparable with other larger habitats. It was 0.5 per 100 square kilometers. The detection probability was 0.018, and the realized abundance in the area that we effectively sampled, which was 460 square kilometers, was 4 individuals with the CIs anywhere between 4 to 9. Apart from snow leopards, we found 16 species of mammals and a high diversity in the region. The lower areas had black bears, rhesus macaques, followed by then moving towards the higher regions where there were brown bears, marmots, musk deer, ibex, red fox, pika. And for their relative abundances, we found that marmots and pikas were detected the most in front of camera traps in all of the camera traps. The relative abundance of ibex was twice as that of snow leopards. And species like the musk deer, which are highly elusive and highly rare, had the lowest relative abundance in the landscape. There were also leopard cats, martens, stoats. Right. So what did we learn from our surveys, and what are the major messages? So this area, Kishtawar High Altitude National Park, it's a significant snow leopard habitat, and it's also connected from our ongoing work. We are able to say this, that it's also connected to other key populations in the landscape and in the region of Jammu and Kashmir. The marmots, long-tailed marmots, could also be an important potential summer prey. We need more research to inform that for this landscape, but we know from other work that this has been the case. Because the footprint of livestock and herders in the landscape is also quite high, although it's seasonal, there is potential of human-wildlife interactions, both negative and positive. Also, what is important is that we keep working alongside the locals, and this baseline builds up, and we continue monitoring snow leopards and their populations in this landscape and in larger connected regions. As I was mentioning, this in the entire region of Jammu Kashmir, when we tried to plot the habitat distribution, we also see that Khamp here is connected to snow leopard populations, and it's neighboring to regions of Ladakh and Himachal Pradesh, which are also important snow leopard habitats. So this is a great it's part of a great larger important snow leopard area in the region. Now, building upon the baseline work and taking this work ahead, our colleagues and members of the team are also trying to do a threat assessment exercise to understand and highlight the activities, highlight the gaps in human-wildlife conflict management. And a lot of their findings are then informing the management in the region so that meaningful interventions can be developed to deal with this. Also, it's very interesting to note that the threats or the conflicts are highly different from valley to valley in all of the regions and across the elevational gradient, where the villages are in lower elevational areas, and the conflict there is different with the different kinds of animals found in the region. And in the high pastures or mountains, the conflict therefore varies. Also, another interesting thing from the work is that now 2 of our locals who have been playing significant role in the data collection and camera trapping work for snow leopards are now partnering with the forest department and continuing the monitoring work in the landscape for this population. Yes. With that, I would end the talk, and I like to thank everybody for patiently listening, and thank Integrative Conservation and Snow Leopard Network also for inviting me to give this talk. Thank you so much.
Okay, thank you, Charu, for your great talk. Ricky, we're a little bit earlier. Should we ask questions now or later to ask questions together?
It's up to you. Do you want, Charu? Would you like to answer questions now, or we can do it at the end?
Anything is fine.
Okay.
Yeah, I want to ask Archana Bahuguna.
Yeah, great. Yeah, please.
Yeah, you know that the prey base of snow leopard is also Palash cat. So did she encounter any Palash cat during survey?
No, we did not encounter Pallas's cat in this part of the region. I'm not sure if they have been documented from Jammu and Kashmir yet.
Any other questions?
Yeah, I was just wondering which techniques or tools you use to do the identification of the individuals.
Right, because a lot of these times snow leopards fur is muffled and it's difficult to use softwares. We manually identified the individuals. So two people independently look at the data set, identify, and then their profiles are matched or merged. Discrepancies are then resolved by a third observer. So it's we followed the two-step process to identify snow leopards. Yes. But all the people identifying snow leopards were trained using the CAM training model, the web link that has been set up to aid researchers in identifying snow leopards.
Okay. So if in the future you had the ability to, if you like, expand and have quite a lot more data that would make doing it manually harder, would you just get more people power to still do it manually, or you're hoping, how do you find a way around it?
Right, that's a very interesting question. I really hope that we're able to develop or get a model to train that way. I feel if it's long-term work and if you have similar flanks or similar body parts, maybe there are some models who perform to a fairer extent, but still a human ID or a human verification is needed. At the moment, we do it manually only.
Yeah. And a slightly different question, if you don't mind. What sort of effects of climate change are you starting to see, like both in terms of when you talk to the local people and perhaps how they are using the habitat differently? You know, are they going further up the mountain? Is the season extending or shrinking? And that by, what effect then does that have on the snow leopards?
Right. That's a very, very interesting question. So from a lot of these threat assessment surveys, what has come out and stood out is that people are complaining a reduction in the water, or there's less water, at least in the high mountains or those areas. And a lot of these glaciers are melting at a faster rate. So what we suspect is maybe then in winters, the snow leopards would come closer to villages, or these other wildlife is what they were complaining largely, because the villages are way down in the valleys. They were complaining that those wildlife might want to come near the villages or those areas, and that would lead to more conflict in that sense. And of course, the seasonal, like we've been keep seeing that snow is getting delayed in the landscape. It's more erratic, it's more abrupt, and that way seasons have been changing drastically there.
Yeah, great. Thank you so much.
Thank you.
Okay, there's another question from Chiwi. In which season the survey was taken? Any relation to the movement of the snow leopard with the seasonal movement of the livestock herders?
Yes, that's a very, very nice question. So when we did it for this research for first time, we did it in early April to June. So that's like end of April to June, where there's late spring transcending into the summer. And the herders use the landscape from end of May to September. So we tried covering the landscape when it was slightly accessible, but there was still snow in a lot of these areas to try maximize getting snow leopard captures in the region, and avoid the disturbance of the herders and the large amounts of livestock coming in. But of course, we did see, because the last 20 days of our camera trapping period did encounter a lot of livestock. So learning from that, now we survey in autumn, from September to November, that period, to avoid completely detecting, to avoid the period where livestock and herders are there. And it is interesting to note in parts of the neighboring regions that you do get more snow leopards when it's completely, when we are placing camera traps when the herders are not. using the area.
Okay, another question from
Right.
Sanjay. Yeah. Did you use 2 camera traps in single location to capture both flanks of the snow leopards?
No, we used 1 camera trap only, but we tried placing it in places where they spray or they mark their territory. So in tighter cliffs, so there you would expect the animal to turn around and maximize getting 2 flanks, both the flanks of the individual. And otherwise, if we are aiming to place it in nearby areas or in similar grids, then we would, if you're suspecting the animal to walk that path, you would change the position to try and get both the flanks.
Okay, another question from Flavia. Your research also monitors other mountain mammals based on your baseline data in Kishwa. How are the current population dynamics of wild ungulates influencing the spatial distribution and carrying capacity of snow leopards in that specific landscape?
Yeah, thank you. Thank you so much for this question, Flavia. This is also very interesting and very important. So our work in the larger region of Jammu and Kashmir informs us that the primary prey, that is ibex in the region, is found in most of the part, but snow leopards are not. So that we suspect would require more research to answer. And we have not yet monitored or studied the population dynamics of prey in the region. But yes, that is something we also are interested to look forward. Like, how are these predator-prey dynamics playing out in that landscape? Or if there are these areas where ibex is still found, but snow leopards are not using the landscape. So what could be done in that sense?
Okay, great. Thank you, Charu, so much.
Thank you so much.
And okay, Charu just gave a great talk on our first theme of this special issue, establishing baselines. And next, we will invite another speaker, our second speaker, Chen, who will. So I'll give a very brief introduction of Chen. So Chen is in charge of China Nature Watch program of Shanshui Conservation Center since 2019, and focusing on and dedicated to promoting the mainstreaming of biodiversity conservation in planning, investment, environmental assessment, and corporate behavior through multiple perspectives, including biodiversity database, technological empowerment, scientific research, and policy advocacy to protect biodiversity-sensitive areas from economic development. And today, Chen will present her paper titled Next Generation Sequencing of Fecal DNA, a Novel Insight into the Metagenome, Phylogeography of the Snow Leopard. And yeah, Chen, the floor is yours.
Yeah. Thank you very much for SLN's invitation to give me this opportunity to share my published paper in my very early career. And thank you for Juan's introduction. Actually, I was a colleague of Juan, and so you will see Juan's name also in my PPT, because she has been contributing a lot of sampling for this study. Thank you very much. So today I'm very honored to be here to share my just published paper last year, Next Generation Sequencing of Fecal DNA, a novel insight into the metagenome phylogeography of the snow leopard. Actually, I'm very excited to discuss how we use this new genomic tool to unlock some secrets of snow leopard, because everyone work on snow leopard genetic study. We know that how difficult that we can get samples, especially if we want to do the population scale or larger scale sampling. only scats can do the best, but we cannot expect tissues or blood. We have no such samples, so that's why we wanted to explore some new pathway to those genetic information. So this study actually have been worked on for many years. And thanks to so many my colleagues in Peking University and the Shanshui Conservation Center, so that this study can finally came out. Actually, not only colleagues, but also this work has been supported by a lot of local governments. And these are two photos of the Peking University Center for Nature and Society, the group, and also the country's Shanshui Conservation Center colleagues. So let me start with why this study matters. Actually, snow leopard distributed across 12 countries, but in China, we are holding approximately 60% of the species' global habitat. So there are several conservation genetics research published all around the global habitats, and a few are about Chinese habitats. But most of them are central for identifying the genetic diversity. Why? Here is the core challenge in such a study. Like snow leopard are extraordinarily exclusive in collecting tissue samples from wide individuals at a population scale is simply not practical. And fecal samples offers a non-invasive alternative. Field teams can find scats along transects without ever disturbing the animals. And it's also very more possible when we just when we collect samples during our field work. And one problem is that fecal DNA is highly degraded, typically fragmented into around 300 base pairs and contains very low quantities of endogenous DNA alongside large amounts of microbial DNA. Based on next generation sequencing, we call that NGS, however, is uniquely suited to exactly this kind of short and degraded material. So this alignment between the technical characteristics of fecal DNA and NGS is, as we will see, at the heart of our approach. Previous studies uses PCR to look at short segments of mitochondrial DNA, but these short segments often were lack of genetic diversity. Some studies even suggested that there was almost no variation across the entire range, which we suspected was maybe it was a technical limitation rather than a biological reality. So our study set out to address this directly with two objectives. First, to try to establish a robust NGS-based workflow for assembling complete snow leopard mitogenomes from fecal DNA. And second objective is to reconstruct the mitogenome phylogeography of wide populations in the eastern Qinghai Tibet Plateau, one of the world's largest continuous snow leopard habitats at approximately 490,000 square kilometers, covering Qilian Mountain, Sanjiangyuan region, and Henduan Mountains. So these are where we did the sampling. Actually, Juan just mentioned Dr. Xiaolin Yun, and she is also in this photo. This is our in Sichuan of China, and this is in Zaduo, in Sojia, and also in Sichuan Ganzi. So this is our workflow. There are 5 steps. First, we collected, actually, we have collected hundreds of samples during our field work, but we only chose 19 of them, which has been, we already identified the species and also individuals so that we have all of them as independent samples. These are all they were collected during the field transect surveys in Qinghai and Sichuan provinces between 2014 and 2019, covering 3 major geographic units: the Qilian Mountain, the Sanjiangyuan region, and the Hengduan Mountains. Samples were preserved in 95% ethanol and stored at -20 degree. Second, DNA were extracted using a very standard commercial kit. And third, library construction. This is where our key modification comes in. We simply skipped the ultrasonic fragmentation step that standard protocols include, because fecal DNA is already fragmented inside the body to approximate the size range around 300 base pair, which aligns naturally with the NGS shotgun approach. Fourth, we use the Illumina Nova system. We generated about 6 to 12 gigabases of raw data per sample in 150 base paired end reads. And fifth, and finally, we extracted mitochondrial reads by mapping to a reference mitogenome, applied quality trimming and removed duplicates, and assembled a circular mitogenome contig for each sample, and use Mr. Bayes to do the analysis of the phylogenetic reconstruction. And for our results, first of all, we have successfully assembled 17 of the 19 fecal samples. Success rate is nearly 90%, and 2 samples failed because one due to insufficient coverage, and the other one due to nuclear mitochondrial feed-on genes, which can confound metagenome assembly. Each metagenome was approximately 16,720 base pairs in length, and across the 17 sequences, we identify 67 SNPs. An important practical note is that the coverage depth varied enormously across samples, from as low as 7-fold to as high as 3,500-fold, reflecting the highly variable indigenous DNA content typical of fecal samples. We found that generating approximately 4 gigabytes of raw data per sample is a cost-efficient threshold. To our phylogeographic finding, our Bayesian phylogenetic analysis revealed that there are 2 divergent mitochondrial lineages in the eastern Qinghai Tibet plateau, shown here on the map on the left. For the green triangle, this is lineage A. It is broadly distributed across the Zhenjiang region and the Hengduan Mountain on the south part of our study area. And the blue circles, which is the lineage B, is concentrated in the Chilean mountains in the north part of our study area. And this lineage seems to be a more ancient, distinct clade. And in the middle, there is a place named Maqing. Both lineages co-occur in Maqing. This suggests that we are looking at a north-south phylogeographic slit, which a zone of secondary contact in the middle. And this slide shows how this divergence are significant. The average genetic distance between our two lineages is 0.31%. This level of differentiation is comparable to what we see between major subspecies of lion and tigers. It also suggests that it might be a very ancient divergence, perhaps dating back to 50,000 to 80,000 years. This level of divergence just appeared in tiger and lion. It's subspecies divergent, but in snow leopard is only between populations. And we also observe some mitonuclear discordance. That means the nuclear genetic differentiation among these populations is much lower than the mitochondrial DNA divergence. So this pattern is common in cats and is consistent with Asian divergence of maternal lineages, followed by more recent male-biased dispersal and admixture. So this is we worked on why this workflow works. First, this fragmented fecal DNA size and this NGS, they matched. And second, the high copy number of mitochondrial DNA makes this work because we always find enough, even if we have very low concentration of DNA, but we still have enough mitochondrial reads to do an analysis. And third, perhaps most importantly for the field, this workflow uses standard commercial services, generates data to NCBI in directly comparable formats. And it is very useful for cross-laboratory comparability, so that if anyone here used to use the microsatellite-based study, you will find it is so difficult to compare between or among different labs. But this new tool or new method can just make that happen. For conservation, the implication is also very significant. The distinct mitochondrial lineage we identified suggests the Chilean mountain population may have been representing a historical isolated evolutionary unit, potentially just dedicated conservation management separate from Sanjiangyuan and Hengduan population. And it's also, we are on the big background of climate change currently. So this Xilian Mountain used to play a role of climate refugee. In the future, we will expect that that place play the role in the future. And also for the transboundary study, it is very important because we have identified one Mongolia sample within the lineage A, which is mostly covering Sanjiangyuan and Hengduan mountain samples, which are on the north. So there might be some connections between them. And also the mitonuclear discordance we observed should inform how we design connectivity conservation. But of course, this study is very preliminary one. So this snow leopard male biased dispersal requires further more evidence. So looking ahead, our study is the starting point. It is geographically limited to the eastern Qinghai Tibet plateau. That was because we wanted to do a quick one so that we can just share this a new method to everybody and then to break the limitation that is always just stopping us from a collaboration or comparing from different teams. So expanding this workflow to cover all 12 range countries is our dream. Hope this new method can help with that. Then at the end, I want to summarize. In this study, we established a robust and cost-efficient workflow for assembling complete snow leopard mitogenomes from fecal DNA, which allows huge-scale genetic studies. So we also reviewed 2 divergent lineages with north-south slits in the eastern Qinghai Tibet plateau at the level of divergence comparable to recognized subspecies of lions and tigers. And we preliminary identify the Qinghai, the Xilian Mountains as a likely glacial refugee with unique evolutionary significance, which may need more care, more notice, and special management. So that is all for my presentation and sharing of my paper. Thank you very much, and I hope we can just discuss about this new method, and all your questions are very welcome.
Okay, thank you so much, Chen Chen, for the great presentation. I saw many people has already raised their hand. You could directly ask the question or type your question in the chat. Okay.
Yes, I have a question. Like, how close is Mongolian population to Chinese population?
Pardon? Oh.
Like on a nucleotide basis, what do you think that Mongolian population is very close to Chinese?
Oh, you mean they're genetically close?
Yeah, genetically, whether they are very close, then how much you find that, and how distant it is from India?
Oh, because we don't have Indian samples, or we did, and we also didn't find any just information or segments, just reads on the NCBI. So actually, we have no idea if Indian population have close or far just distance from Chinese population. But for Mongolia, they have we found several reads online, so that's why we can compare. And according to our results, actually they're mostly close to the Chilean population, which is in lineage B. But one of them was close to lineage A. So there might be some mix, maybe not through Chilean, but through other pathways. But we don't know because this limitation of our samples and actually the mitochondrial information is only for this gender biased because of the gender biased marker of mitochondrial. So it can only show one side of the mirror. If we want to just know more, we need the nuclear DNA to provide more information. But as I can I can make a guess that if we put more Mongolia samples together with Chinese samples, we will see a lot of interesting things happening. And one question, I also have something that I want to find out. We found the NCBI Mongolia sample DNA, but we didn't find out where it was sampled. And we tried to ask people to find out, but no one can find out that for us. So maybe through our network, we can just find this information.
Nice, thank you.
Okay, Neil, you could ask your question, please.
Thank you. Thank you, Jen. Can you hear me? Can you hear
Yes.
me now? Can you hear me now? Yes.
Yes.
My question is a very interesting presentation, but my question I wanted to find out more from you is if you can tell us what is the divergence in snow leopard lineage? What does it tell us about the impact this has on geographical conservation needs of snow leopards? How does what we are studying from this presentation, how does it impact the geographical conservation needs of snow leopards? If you could tell us a bit more about that, please.
Thank you for this question. Yes, the genetic study is often very far from our conservation. And if you want to just figure out how that can help with our management, on ground management, I think if we only just study on how high is the genetic diversity, we will never find out which population should be prioritized. But like in our paper, we were very lucky to find a very ancient clade, which is maybe just as an example, the Chilean mountain population might be relatively independent unit from other populations. Then we have been talking with the Chilean mountain to let the management bureau to know the importance of their population. And we also were just pushing them to figure out if this kind of maybe the resources, the funding can go more into Chilean mountain, because that is the refuge or maybe the future place that preserve this ancient lineage. So that's what we do. But if you are talking about how we can just use this genetic results to do some management. I think there are still some distance between understanding distance between them. So maybe we need more just people to who work on genetics and work on ground management to talk together so that we can find out. It's very hard for me to answer this question, but it's a very good one. Thank you.
Thank you.
Okay, thank you. Sanjaya, you could ask your questions.
I have a couple of questions, some methodical questions. The first one is how old the samples that you use for the extraction of DNA? Did you use any very fresh scat, or did you manage to extract DNA from old scat as well? And the other one is, did you run any phylogenetic analysis on nuclear DNA as well? And if so, did you see the same pattern as mitochondrial DNA? And the other one is kind of a comment. I think the nuclear mitochondrial discordance has been observed in many conditions where the evolution rate in mitochondria is much higher than the evolution rate in nuclear genome. So I think if we can add more nuclear genome to the tree, then it can resolve, or it can give us more realistic tree than the mitochondrial trees. Yeah, those are my questions.
Thank you. Thank you for your comments. Yes, you see this this paper is a very just quick paper. So I think it's the core of our just sharing is this new method, and we saw the difficulty of having more genetic information into analysis. But if you have you followed with the snow leopard population, genetic population study, last year there was another paper published who has done has worked on the whole nuclear DNA. So I think if we can combine this two, but of course that will cost more. So this mitochondria is more just much more much less expensive than that. But I think this new method can help more people to work together, and not only on nuclear but also on mitochondrial. And these two papers can help people can have more labs to collaborate. And for your questions, the first one is, did we extract DNA from all samples? Is that?
Yeah, that's the question.
Yeah, of course. We did our best to extract DNA and do the species identification and individual identification as much as we can do. So almost all the samples were just managed operated in the lab. And we have a database to store all this information. But in this study, we only used 19 samples from them. Yeah, and the second one is, I forgot about the second question. Sorry.
The second one was, did you run any nuclear tree
Nuclear
to
tree.
compare with the mitochondrial tree?
Actually, my colleague might be my colleague had been doing that, and I haven't did that because we only use the microsatellites to also figure out this genetic structure around this region. And if you are interested, we can just I can share some information through email or something. It's the result hasn't been published yet.
Yeah, thank you very much. I'll write to you.
Okay, thank you for your question.
Okay, there's another question in the chat from Alina. And she asked, were there any limitations to using ethanol for preservation in the field? Did you consider using other methods of fecal preservation?
Mm-hmm. Limitation to using. Actually, we also use that, what's that? Silicone, but it's not as good as ethanol. Actually, if you use ethanol, you need the 99% ethanol. It's not those you can buy in the what's that? In the store. Usually you need to buy the very, very pure ethanol so that it can extract the water from the sample. But that is only for if you can do the sampling and very quickly transfer those samples to your lab to the fridge, minus 20 degree fridge, then the ethanol is not that much important. But if that will take a lot of time, a lot of days, then I will strongly suggest the ethanol to be the best choice.
Okay, another question from Alina. You mentioned that the divergence between these lineages is comparable with differences between subspecies of other large cats. Is there thought to designate snow leopard subspecies? What would be the pros and cons of doing that?
I think we have to involve the nuclear genome.
Okay, last question from Jimmy. Is this method applicable to study snow leopard seasonal dietary preferences?
The NGS, the next generation sequencing, can also do the dietary analysis. That's for sure. Our team and collaborator has already published 2 papers about that, using this very similar method based on NGS. But the methods actually are very, very different from just my paper and that dietary analysis. If you want to do that, that will have to based on metabolic coding, which that means you need to do more PCRs so that you can have some specific segments from the DNA, and then to do the species identification, and not only for one species. You need to use the NGS to do the multi-species identification at the same time. And then the analysis methods and the software you use are very, very different. But if you're interested, I can share the paper with you. We have been working using those methods on snow leopard and a lot of species in the same habitat, like red fox, Tibetan fox, and other species, other carnivores. Yeah, the result was also very, very interesting.
Okay, thank you. Thank you both, Charu and Chen, for your wonderful presentation today. And thank you for everyone in the audience for your great, thoughtful questions and engagement.
Thank you so much, Joanne. Thank you, Ahimsa, Jia, Charu, Cece, and everyone at Integrative Conservation for giving us this webinar. We look forward to the next one, and I hope you guys can all join us. And it was great to see so much of participation and engagement, and we look forward to the next one. Thank you.
Thank you.
Thank you. And I encourage all of you to access the full special issue. All the papers are open access. So if you have more questions, you could read the papers and directly email the authors. Okay, thank you.
Thank you.